Today I will describe to you about the steps involved in gene cloning. It involves 5 different steps. The first step is the isolation of the target DNA and cloning vector.
In previous video, I have already described to you about the first step, which is isolation of target DNA and cloning vector. So in this recording, I will describe to you about the second step, which is to cut both DNA with the same restriction enzyme.
The second step is to cut both DNA with the same restriction enzyme. So let's say your fluorescent gene is gene D, but you actually cannot see because
they are very small. We cannot see them with our naked eye.
So this foreign DNA will be cut by the same restriction enzyme in order to get DNA fragments. So let's say here we have the restriction site for EcoRI, GAATTC. So EcoRI can cut in between the guanine and adenine in this recognition sequence. So if you have several recognition sequences, then the EcoRI will cut at this site. So when it cut at this site, it will cause the formation of the DNA fragments. So when it cuts, it actually cuts at the phosphodiester bond and causing the DNA to become fragmented. So you do the same about this process to the cloning vector that you used. So in this discussion is the plasmid. So this is the multiple cloning site of the plasmid, in which in this site it can be cut by various types of enzymes. So let's say it has the GAATTC, the recognition sequence for EcoRI. That means it will cut at this region and also this region. So you note the palindromic sequence of the restriction site. The GAATTC is read the same on the double-stranded DNA on opposite orientation, GAATTC. It allows the restriction enzyme to cut at both strands. So once it cuts the phosphodiester bond, it can only be held by the weak hydrogen bonds, which causes the plasmid to become open. So the digested target DNA with the digested plasmid is mixed together. So you actually have lots of DNA fragments. And your genes can be in multiple copies.
Both fragments of DNA is mixed together with digested plasmid in step 3. We will continue our discussion in the next video, which is recombinant DNA step 3. See you again!